So let us talk about cell injury and cell death. Okay. So basically, we're going to talk about when we talk about cell death, we're going to talk about two types of cell death, which is necrosis and also the apoptosis. So we are going to consider the mechanism of this. So first, let's start with cell injury. So in the previous video, we talked about cell adaptation. Okay, so cell injury comes as a result when the cell fail to adapt to a stimuli. Okay, so when the cell fail to adapt to a stimuli, it causes injury to the cell. This becomes when the stimulus is mild. Okay, is mild. So cell injury, it can be in two forms. It can be the reversible, versible, and the irreversible cell injury. The reversible cell injury, this simply means that when the cell becomes injured, meaning it can go back to the normal, after the stimulus is removed, or after the body the supply of the cell requirements becomes back to normal. So the cell can be reversible. So when it comes to irreversible, this means the cell can't reverse. It can lead to cell death. And this cell death, it is in two types. The cell can die by apoptosis, or the cell can die by necrosis. So for the cell to undergo irreversibles, this means that the stimulus is very severe. And then for the cell to undergo the reversible, it means that stimulus is mild. So now let's first start with necrosis. Let us start with necrosis. Okay, what is the main cause of necrosis? The main cause of necrosis, it is the hypoxia. What is hypoxia? Hypoxia, it simply means the decrease in oxygen supply to the cell. This is the normal cell. Okay, I've just depicted the organelles that we will consider that contribute to cell injury and death. So this is the nucleus, this is the mitochondria, and this is the ribosome in the present left in the present reticulum. So the left in the present reticulum, endopresent reticulum. So it is the left in the present reticulum with the ribosomes attached to it. So the main cause of ischemia, it is the main cause of hypoxia, it is ischemia. Okay, what is ischemia? Ischemia simply means the decrease in the blood supply. Decrease in the blood supply. We know the function of the blood is to carry oxygen. So now, when there is a reduction in the blood supply to the cell or to the tissue, what can that lead to? It means that it will also reduce the oxygen supply to the tissue. Okay, so now, what is the function of oxygen in the cell? Okay, let's say there is oxygen here. Oxygen. We know that oxygen is very much important in the production of ATP when it enters the mitochondria. So now, when there is reduced supply of oxygen, it means that there will be also reduced supply of ATP. Why the reduction in ATP can lead to cell injury? ATP is vital to regulate the function of the sodium potassium pump. Okay, sodium potassium pump, this pump. This means that without ATP, this pump, it can't function. What is the function of this sodium potassium pump? This sodium potassium pump, it pumps potassium, 2 potassium ions into the cell and 3 sodium ions out of the cell. This is the type of active transport, which is part of osmosis. So what you need to understand about sodium and potassium is that outside the cell, sodium is more concentrated. Then inside the cell, potassium is more concentrated. So that's why this one is moving outside the cell through the process of osmosis, but it is the active transport type of osmosis, because it is moving to from the low to high concentration. So now, let's say when there is the reduction in ATP production, what will result? Meaning, it will decrease the function of the pump. It will decrease the function of sodium potassium pump. This means that the normal amount of sodium that leaves out of the cell and the normal amount of sodium that leaves into a cell per given time is disturbed, meaning this movement decreases both the number of sodium that is entering into the cell and the number of potassium entering and the number of sodium that is entering. It is decreased per given time. So now, what will come as a result when there is a reduction in this processing? We also need to know that in the cell here, sodium enters through sodium channel. It enters through sodium channel, but gets out of the cell through sodium potassium pump. Okay, so now, if there is a defect or the reduction in the normal function of this pump, this simply means that sodium will continue entering. Okay, sodium will continue entering, but there will be a reduction in its moving out of the cell, such that you will find that there will be more accumulation of sodium inside the cell. This leads to sodium influx, which is the sodium ion influx. Okay, this leads to the sodium ion influx. This sodium ion influx, what does it cause to the cell? It depolarizes the cell. When it depolarizes, means the cell becomes more positive. When the cell becomes more positive, what does it cause? It causes the opening of the calcium channel. Okay, the calcium channel opens. It stimulates the calcium channel. When the calcium channel opens, what will result in the entering of the calcium channel? On the calcium ion. When the calcium ion, meaning the calcium ion will increase inside the cell. Calcium ion increases inside the cell. What these calcium ion cause inside the cell? This calcium ion, it will act as the cofactor. Okay. Before we say what this cofactor, which is the calcium ion, do, let us go back to sodium. As the sodium is increasing here, it also pulls water to enter into the cell, water also follows. As water is following, what does it cause? It causes the increase in water in the cell, okay, in the cell. This causes the cell to swell, okay? The cell swells as water is accumulating. So as the water is accumulating, this water, it causes the what? The detachment of the ribosome. Ribosome detaches, okay? It causes the detachment because it also causes the endoplasmic reticulum to swell. These ribosomes, they detach from the ER, okay? As the ribosome detaches, meaning this reduces the protein synthesis, okay? As the protein synthesis reduces, what does it mean? It means that there is a reduction of the proteins that makes the cell membrane of the cell, and also there is a reduction in the cytoskeleton of the cell. So that's cell injury. That is how the cells starts to become injured. Next, what is this calcium, which is act as the cofactor? Cofactor, it activates the enzyme after binding to it. So calcium acts as an activator, enzyme activator. It activates the enzymes, okay? What are those types of enzymes? There is proteases, okay? Phosphor Lipids enzymes, Antipaase enzyme, and endonuclease enzymes, DNA enzyme. So these enzymes are activated by calciums, and the same calcium causes them to become activated. So when these enzymes are activated, let's start with proteases. These proteases, what do they do? They break down the cytoskeleton of the cell. When the cytoskeleton of the cell is broken down, what is the result? Meaning the cell, it loses its stability, it loses its integrity, it becomes weak, such that it can die at any time. When we talk about phospholipids, these phospholipids, it breaks down the bilayer membrane, which is made up of the phospholipids. When this bilayer is broken down, it breaks the bilayer. Also the bilayer of the mitochondria, it breaks down the bilayer of the mitochondria. So when the membrane of the mitochondria is being broken down, what does it mean? Meaning it leads to reduction of ATP synthesis here. Listen. So when ATP, meaning it causes another reduction in ATP. Since initially say oxygen, when there's less oxygen level, it means that there is a reduction in the ATP production, because oxygen is required for glycolysis to take place so that ATP can be produced. So when there is a reduction in ATP production, it means that the sodium pump can't function. Listen, there is a reduction in the function of the sodium pump. When there is a reduction in the function of the sodium pump, it means that sodium here will continue entering into the cell, but it won't be going out of the cell. It won't be going out of the cell. As this sodium is entering the cell, meaning it will depolarize the cell will become depolarized. As the cell becomes depolarized, meaning it will become more positive. As it becomes more positive, this means that it will activate the opening of the calcium channel. When the calcium channel opens, it allows calcium to enter into the cell. As calcium is entering the cell, it causes the concentration of calcium, because this sodium to continue opening the calcium channels. This increases the calcium ion influx, and also since sodium is entering, it will cause sodium ion influx. So this calcium, it acts as the cofactor which activates different enzymes of the cell, such as the proteases, phospholipids, adipase, and the nuclease. So these enzymes, that's like protease, it breaks down the cytoskeleton. Then phospholipids, it breaks down the membrane of the cell, the membrane of the mitochondria, and also the membrane of the vesicle of lysosomal vesicle. When this membrane is broken down, what leads to? It leads to the production of lysosomal enzymes. Listen, when these enzymes are activated, including the antipase, it is responsible for consuming ATP. ATP is already reduced. Now, if this enzyme is activated, meaning it will consume even the little ATP present, which can also lead to severe result, the injury of the cell. In the nucleus, it breaks down the nucleus, and we know the nucleus is very, very much important to the cell, and also the DNase. So when these enzymes do the damages of this cell, this is leading to cell injury. The cell becomes injured. But now, what determines if the injury is reversible or irreversible? It is the type of stimulus. Is it severe or not severe? So let's start with the injury that is irreversible. For the reversible means that the stimulus is mild. Have you seen? The stimulus is mild. For it to become reversible, it means that the stimulus, let me just wrap here. The stimulus is mild. So when the stimulus is mild, this means that at least there is enough oxygen to produce the large amount of ATP. When the ATP is produced, it means that at least this sodium potassium pump can work, but it is just reduced. This also means that sodium can be entering, but also it can be going out, meaning it won't cause much of the calcium channel to open. So meaning as much calcium channel is not opening, meaning small calcium is entering. As small calcium is entering, it means that less enzymes will be activated at the least amount. So this will only cause the small damage to the cell. Okay, it will only cause small damage to the cell. So when this occurs, meaning those, you can see the cell forming some blobs, okay, like this. So some blobs, you can see them. So now, when the stimulus is severe, meaning the reduction of oxygen is so severe, it means that even the ATP production will be very, 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 very minimal, very small. When the ATP production is very small, meaning this sodium potassium pump will be inhibited so strongly, meaning it will want to work because there is very, very little ATP. When this won't work or inhibited completely, meaning sodium will keep on entering the cell, but no sodium going out. As it is entering, it causes depolarization. As it is causing depolarization, it causes calcium channel to open. As the calcium channel opens, more of the calcium ion will enter. As it enters, it will activate these proteins, which is the proteases, like phospholipids and the nucleases. These are activated. These proteins, the enzymes, they cause severe damage to the cell. As it causes the severe damage to the cell, this leads to irreversible cell injury. And this irreversible cell injury, since there is too much damage to the cell components, this is a death. With the type of death, it's a necrosis. So necrosis simply means a cell to die, the death of the cell due to the abnormal condition, due to the abnormal requirement of the cell, okay? The reduced requirement of the cell or the abnormal chemicals that the cell is subjected to. But the main cause is the hypoxia. Even some chemicals can cause the cell to die, can cause the injury, but the main cause is hypoxia. Hypoxia simply means a reduction in oxygen. When there is a reduction in oxygen, what causes the reduction of oxygen is just the decrease in oxygen supply to the cell. So this is necrosis. Okay. So now let's talk about the apoptosis. Before I talk about apoptosis, what you need to know is that the process of necrosis, it is as a result of pathological cause only. The only thing that causes is the pathological causes. But for apoptosis, there are two, pathological and also physiological. So now let's talk about the apoptosis. How does it come about, or the mechanism? Let's talk about apoptosis. So apoptosis, it is just the cell programmed death. Cell programmed death. What does this mean? It is the normal process by which the cell undergoes the death. But also, this can be normal, but it can also be abnormal. Normal and abnormal. Okay. Abnormal and normal. So when we talk about abnormal, we are talking about the pathological cause. When we talk about the normal, we are talking about the physiological cause. So let's talk about the pathological cause. Pathological cause of apoptosis. So when we talk about the pathological cause of apoptosis, it can be as a result of the DNA damage. Okay, DNA damage due to some chemicals, and also it can be as a result of the protein misfolding. Okay. It can also come as a result of the cell death due to viral infections, okay? Due to the viral infections of the cell. So this is the pathological cause. Okay. Let us go to physiological. The physiological cause of apoptosis, which is the normal cell-programmed death for physiological. The causes of physiological are during embryogenesis, okay? Embryogenesis. Then another one, it is due to hormonal withdrawal. Hormonal withdrawal. Then the next one, it is the removal of self-reactive lymphocytes in thymus. So killing of self-reactive lymphocytes, lymphocytes in thymus. Okay, so these are the causes. The first one is for the pathological, the second one is for the physiological. So the pathological, it is when the DNA becomes damaged, protein misfolding, and also the viral affected cells. They undergo the process of apoptosis, but it is a pathological cause. Then we talk about the physiological. We talk about the what? During the what? The embryogenesis life stage, as one is developing, the development of our body. This means that during the development of the body, there are those cells that are being produced. When they are being produced, meaning some they become misfunctional to the body. As they become misfunctional, meaning for them, they can't continue being in our body, meaning they has to be killed. So these cells, they are killed by the process of apoptosis. And also when we talk about the hormonal withdrawal, we're talking about the breakdown of the endometrium, which leads to menstruation, and the breakdown of endometrium due to hormonal withdrawal, which is the estrogen. The other one, which is the killing of self-reactive lymphocytes. This is during thymic education, where the lymphocytes are tested to see its function. Is it able to recognize the fallen cell? But now, when these cells fail to recognize the fallen cell, but instead it recognizes the cells and kills it, this cell can be killed by apoptosis. So these are the causes. Now let us go into the process of apoptosis, which is the mechanism. Okay, so let's get the cause. Now there is a causative here, a cause, mechanism, a cause. You can get any of the causes that we talked about. So let's just put us there is a cause here. Let's say there is protein misfolding or DNA damage as a cause damage. So when this occurs, the first thing there is the first protein. There's a protein that is sense this change, which is known as apoptotic sensors. These apoptotic sensors protein, they sense the damage. Okay, now they sense if the damage is severe or mild. Meaning when it is severe, it activates certain proteins that kills the cell by apoptosis. But when the injury is not, when the damage or the stimulus is not severe but mild, meaning it will activate the certain proteins which inhibit the process of apoptosis. So what are examples of these apoptotic sensors? Examples of apoptotic sensors, there are bead, buds, beam, okay, the beam, okay, bead, bump, okay, and also the puma. These are the apoptotic sensors. These apoptotic sensors, they sense if the stimulus is severe or mild. In this case, we're talking about the damage or the misfolding. When it is severe, what does it do? It activates the pro-apoptotic proteins. Pro-apoptotic proteins. So when the injury is severe, these apoptotic sensors, they activate the pro-apoptotic sensors. What are examples of these pro-apoptotic proteins? Is the backs and the back. Okay, these are examples. But when the injury or when the causative, the cause is mild, meaning this apoptotic sensor, it will activate the anti-apoptotic proteins. These anti-apoptotic proteins, they inhibit the process of apoptosis. To say, so whenever the apoptotic proteins are being stimulated, meaning it will inhibit the process of apoptosis, meaning the cell can go back to its normal. Why? Because the stimulus is mild, it's not that severe. But when it is severe, the pro-apoptotic proteins are being activated. So let's start from the pro-apoptotic. Since we say is that when the apoptotic is being activated, meaning there will be no process of apoptosis, meaning it stops from there. Then the cell can go back to its normal. Let's start from pro-apoptotic. Okay, so it starts from apoptotic sensor, apoptotic sensors, okay, to activate the anti-apoptotic proteins. These apoptotic proteins inhibit the sent from here. Then, if there's the damage is severe, then to activate the apoptotic proteins. Apoptotic proteins. So when these pro-apoptotic proteins, of which we gave an example, see, back or backs, when these proteins are activated, what do? They act on the mitochondria. When they act on the mitochondria, they causes the increase in the permeability of the mitochondria. When there is increase in the permeability of mitochondria, what does it cause? This results into the release of the cytochrome C. When the cytochrome C is being released from the mitochondria, cytochrome C, it activates the initiator proteins. The initiator proteins, which are enzymes. Initiators proteins, which are enzymes. So when cytochrome C activates the initiators, basically the types of initiators they are capses 8 and 9. So capses 8 and 9. When these capses are being activated, they too activate the executioner. Okay, they activate the executioner enzymes. So these capses, they activate the executioner enzymes. What are these enzymes? They are also capses. Capses, which is it? 3 and 6. Okay. These executioner enzymes, which are capses 6 and 3, it now activates the cell enzymes of the cells. The enzymes of the cells are being activated, such as the endonuclease. Okay, all these endonuclease, proteases, enzymes, phospholipids. When these enzymes are activated, what do they cause? They cause in the cell. They cause damage. They cause damage to the cell. Cause damage by degrading the cell components. After causing the damage to the cell, you know what it forms? It forms blobs of the cells. The cell form blobs here. When the cell component have been degraded, then the macrophage comes. The cell eventually, after these enzymes are being degraded, being activated, it degrades the cell enzymes, the cell components. It destroys the cell components. Destroying the cell components leads to cell death. So then the macrophage comes. The macrophage. This is macrophage. It engulfs the cell and it destroys it. Destroys the cell. Hence the cell dies. So this is the process of apoptosis and also the necrosis.